Hello, so in this video, we are going to be looking at, you must know, number one, examples of endergonic and exergonic reactions, the role of ATP in energy coupling, that enzymes work by lowering the energy of activation. We're going to look at the catalytic cycle of an enzyme that results in the production of a final product. We are going to skip this one. This is going to be the next video. And then we are going to be looking at how the shape of enzymes, their active sites, and interaction with specific molecules affect their function. And then we'll get back to this last one in the next video. So let's recap the video that we just, or the lecture that we just finished, lecture 6A. So in lecture 6A, we learned that energy is required for life. And why is that? Well, that's because inside a cell, we have all these different organelles, and each of these organelles are kind of like a factory. It's a protein making factory. And in order to make proteins, chemical reactions need to occur, making molecules and breaking molecules apart. So thousands of reactions are occurring, and energy is needed to basically allow those little machines, those little organelles, to function correctly. So we learned about two different ways for chemical reaction to occur. This is called metabolism. Metabolism is all of the chemical reactions that happen in the entirety of your body. So two different types, just to recap. Catabolic reactions is taking a molecule and breaking it down into smaller pieces, and in the process releasing energy. So this is what happens when we eat food. So when we eat food, we have molecules from our food like glucose and carbohydrates, fats, proteins, and the whole reason why we eat this food is so that we can get energy from the food, kind of like a sponge. If you were to take a sponge and squeeze it, you get water coming out. In the same way, a large molecule is full of energy. The bonds in that molecule are full of energy. And if you were to chop it up into smaller pieces, energy gets released, and that energy can be used in the cell to do work, basically to allow the cell to function properly, right? To create proteins and to move around, all sorts of different functions. We also learned about anabolic reactions. So if you were to take small molecules and build larger ones, that's anabolism. Now, where in the cell would we see this happen? Well, if you're building proteins, right? So we took the energy from our food, we broke it down, and that energy now is going to be used to take smaller molecules, for example, amino acids, the monomers of proteins, and through anabolism, we can put those amino acids together and form a larger molecule, a protein, right? So again, we take our food, we break it down, we release energy. The whole purpose is to allow our cells to build new things, anabolism, build larger molecules like proteins that can be used for whatever the cell needs. So all of life is built on these chemical reactions, anabolic and catabolic reactions. The sum of all those is metabolism. So we know that energy first originates from the sun, and the sun is solar energy, but it's captured by plants, and plants have this special ability to convert energy from the sun into chemical energy, or the energy stored in molecules. So the plant will create glucose or sugar, and the plant will use this glucose and it will break it down to supply its cells with energy, right? So the plant, this is called photosynthesis, taking energy, solar energy, converting it into chemical energy in the form of a molecule of glucose. And then it will do cellular respiration. A plant itself has mitochondria, and the mitochondria will take that glucose molecule and chop it up and use that energy for any reaction that's needed in the cell, in the plant cell. So plants do photosynthesis and cellular respiration. We'll come back to that later this unit. And then, so these are called producers. Producers are autotrophs. They make their own glucose. But some organisms, like this giraffe here, cannot absorb energy from the sun and convert it into chemical energy. These organisms, called heterotrophs or consumers, must consume other living things to get energy. So when this giraffe consumes vegetation or plants, the sugars in this plant will now go into the cells of the giraffe, and the sugars will be broken up into smaller pieces. That reaction will release energy, and that energy can now be used to build new molecules in the cell. Now, some organisms like carnivores, right? So this lion here will want to eat meat, and in the meat, there's now sugar in the, so like if this lion here eats this giraffe, the giraffe has sugar in its body from the plants that it ate. And even though the lion here is not eating the plant straight up, it's going to get those molecules, those biomolecules, the glucose, from eating the meat of a giraffe. So then the lion will now be able to break down that glucose and make energy, and basically supply the cell with energy. All right, so energy is needed for life. Now, living organisms, are endergonic systems. Now, we'll come back to this word in a second, but endergonic basically means energy is needed. So you need energy in order for things to function. Endergonic. An endergonic system requires energy to function, right? We need energy for life. Why do we need energy? Well, we just talked about synthesis, making new things, making biomolecules, so taking amino acids and building proteins, reproduction, active transport, movement, temperature regulation. So these are different functions of cells. We'll talk more about this throughout the year, but basically energy is needed for everything in the cell. We did talk about this last chapter, active transport. So if you are moving a molecule from in the intracellular space to extracellular space, or extracellular to intracellular, basically if you're moving any molecule across a membrane against its concentration gradient, it's not diffusing, you need energy to do that. You need active transport. And so where does that energy come from? Well, it comes from metabolism. It comes from our food and breaking down our food to get energy. Okay, so we just talked about metabolism, and once again, metabolism can be forming bonds between molecules. So for example, unit 1, we talked about dehydration synthesis. That's taking two molecules and causing them to synthesize, to come together by taking out water. This is called an anabolic reaction. An anabolic reaction is the forming of new molecules, so the building of molecules. Or metabolism can be breaking bonds, catabolic reactions. And an example of this from Unit 1 is hydrolysis. So using water, so supplying a molecule with water and breaking apart those molecules is called catabolism, or breaking apart. So these are words you're going to want to memorize. And once again, these reactions work together. So when we eat our food, we are breaking bonds. We are hydrolyzing bonds. So if we eat glucose, we're breaking apart the bonds of glucose. And we're using that energy to form new molecules like proteins. All right, so here's some examples. So here we have two monomers, and if we wanted to build a polymer, we would use dehydration synthesis. So remember, you take two molecules, water is released, and now you're building a polymer, right? So this would be anabolism. We're building new things. Catabolism, breaking apart molecules, requires the hydrolysis reaction, right? You're inputting water and you're breaking this bond here, and now energy is released in the system. All right, here's another picture up close of all the bonds. So dehydration synthesis, synthesizing, putting molecules together, and then hydrolysis. Now, an example, connecting this to the real world. So when plant cells are capturing energy from the sun and creating glucose, right? They take that glucose molecule and they're going to put multiple glucose molecules together, or sometimes even another kind of similar form of glucose called fructose. So glucose and a fructose molecule, they're going to bond them together to form longer chains, and these chains can now be stored and used in other places in the cell. So when plants make glucose, they bond multiple sugar molecules together to make bigger molecules. And then when they're ready to use those molecules, they're ready to break them down, they hydrolyze them. So here's our sucrose molecule built from glucose and fructose. And with the addition of water, now you're breaking those bonds. And now these molecules are now chopped up a little bit, and those can now be used by the cell to get energy. Okay, so we kind of talked about this, but some chemical reactions are releasing energy, and anytime you're talking about a reaction that releases energy, we call it exergonic. So X means out or away, so exergonic means release of energy. Memorize that, write that down. So digesting polymers, so remember, a polymer is a long chain of monomers. If you want to break down that polymer into smaller pieces, then that's an exergonic reaction. Energy is released. So anytime you're thinking about breaking bonds or breaking molecules down, you're releasing energy. Now, some energy is required to break those bonds, so you have to put in some energy first, but overall, the breaking of bonds has a net release of energy. So an example, hydrolysis or catabolism, breaking bonds, breaking molecules apart with the use of water. Okay, then some chemical reactions require the input of energy. These are called endergonic reactions, and this is building. So in order to build something, think about if you're trying to build something, it requires energy to do that. And so if you want to build polymers, if you want to take a glucose monomer and a fructose monomer, and you want to build a polymer, you're going to need some energy input, right? And so this reaction is called dehydration synthesis. So, right, you're taking two monomers and you're releasing water, you're dehydrating it, you're putting those molecules together, and you're building something new, anabolism. Okay, so I know there's a lot of different words, they're all kind of connected. So take a moment, review what is catabolism, what is anabolism, what are exergonic reactions, what are endergonic reactions, what's hydrolysis, what's dehydration synthesis, right? So just to kind of review, exergonic and endergonic, you're talking about energy. So you're talking about how is energy coming in or how is energy coming out. Catabolism, you're talking about the actual molecules. You're talking about the actual atoms. So catabolism, right? That means you are breaking apart molecules. Anabolism means you're building. So when you're thinking about chemical reactions, we can think about two things. We can think about what's actually happening with the matter. How are the molecules and atoms actually moving around and rearranging? And you can think about how energy is involved. When you're building things, is energy input or output? When you're taking molecules apart, what's happening to the energy? So that's basically why we have all these different words to describe these reactions. Okay, so when we're digesting molecules, when we are breaking them apart, we're taking a more organized molecule and we're breaking it apart. We're increasing the entropy in the system, right? So we think back to our other video, we are increasing the entropy. We're lowering the energy state. So we're taking a higher energy molecule and we're breaking it down into its lower energy state. We are increasing the entropy. In the same way, when we're building molecules, we're making it more organized. We're putting a more organized structure together and we're putting it into a higher energy state. That's why this is an endergonic reaction. Energy is needed to build a more organized structure to build a new molecule. Okay, so I know that there are so many words and definitions and ideas when we're talking about metabolism, but I just want to assure you, really, it comes down to two major reactions. Basically, you're either taking a molecule and you're breaking it down, or you're taking molecules and you're putting together and you're building a more organized molecule here. So you're either breaking down or building. So what's happening with the energy here? So let's take this example right here. Let's say we take a glucose molecule. Does the glucose molecule have more energy or less energy than what happens when you break it down? Well, let's review. So remember, a glucose molecule has carbons and hydrogens and oxygens. They're all bonded together covalently. And because the atoms, the carbons and the hydrogens and the oxygens are bonded together, they're sharing electrons, right? They have the potential to break those bonds, right? So right for a current moment, those atoms that make up the glucose molecule, they are in a position where they are close together. But they have the potential to change position, kind of like a spring. When you compress it, it has a potential to change its position. So that's why molecules that are more organized like this on top have more energy. So if you were to take a molecule of glucose, we can call the energy or the potential energy it has, it's called its free energy. So basically free energy or available energy. It's how much energy is available, or kind of like think of that as potential energy. How much energy has a potential to be released. So molecules that are more organized, more structured, molecules that have less entropy, that are less disordered, that are more organized, have more energy. Now, what would happen when you break apart this molecule? Well, when you break apart the molecule, you break the bonds, you break the covalent bonds, you're actually going to release energy. Okay, so energy is going to be released from this reaction. And now you have these smaller molecules are now farther apart. Energy has been released. And so you can graph that. So here we have the start of the reaction, higher energy, and as the bonds break, energy is released here. So kind of think as like a ball rolling downhill. Exergonic reaction. So your reactants, if this glucose molecule is your reactant in the chemical reaction, the amount of energy released from the reactants to the products is the amount of free energy available in this original molecule. So when we're thinking about this glucose molecule, we can describe how much available energy or how much free energy by measuring the difference between how much energy the molecule had before the reaction and then how much energy was released at the end of the reaction. So how much energy does the products have down here? And you'll see that these products have less energy. Now, the energy didn't disappear, it didn't get destroyed, it just got released somewhere else. And that energy that gets released is now used to build new molecules. So if you take smaller molecules here, remember, this is more disordered, this has more entropy, and you want to build a more organized molecule, right, with less entropy. Well, in order to do that, you actually have to take in some energy. You're building a more organized structure. So if we were to graph that, the reactants right here on top have less energy, right? So we're starting with less energy. So how do we build this more complex molecule? Well, it actually requires an input of energy. So energy must come in, okay? So over the course of the reaction, you start with your reactants have less energy, and energy needs to be put in. So the difference between the products, the reactants down here and the products is a change in energy, or the amount of free energy. Another way of thinking about it is the amount of available energy. So for this endergonic reaction, it requires energy to build this molecule. Okay? All right. So again, here's some more graphs to help us think about it. Endergonic versus exergonic graphs. So exergonic energy is released. Now notice that you'll see the reactant here. Here's our organized molecule. We start out at like a baseline level of energy. Okay, so this is the baseline amount of energy. So this is the energy of the reactant. Now you'll notice that as the chemical reaction goes, the x-axis time, we're looking at the y-axis, how much energy is supplied or how much energy is released. So increasing energy versus release of energy. So as the reaction goes, the molecule starts breaking apart. Notice that some energy is required before we break it all down. Okay, now we've already talked about this in class, but this is your activation energy. So there's going to need to be some energy put in order to break these bonds, right? There's energy that's required to break the bonds of this organized molecule. So once some energy comes in, and that energy can either come in by heat, or later on in this video, we'll talk about enzymes called catalysts that will help this reaction happens, but you need some sort of input of energy in order to break the bonds. Once the bonds are broken, now energy can be released. Okay, now notice that if the reactants, or if the products have less energy than the reactants, we have now lost some energy. Energy's been released. And we call that measurement Gibbs free energy. So again, on the previous slide, we described this as free energy or available energy. And we can use this little symbol, delta G. Delta in math means a change, and G stands for Gibbs free energy. And we call that negative because we started off at this baseline, and now we're losing energy, we're releasing energy. And so we have a net loss of energy. So loss of Gibbs free energy. So the change is overall negative. So the starting amount of energy minus the final amount of energy is negative. Okay, so if we started at 0 and we ended at less energy, negative number. Okay, now on this side, endergonic reactions, we start with our reactant has just a baseline of energy, and the products now have more energy. So in order for this to work, you need some energy to be put in. Now we call these reactions that are exergonic, that release energy overall, as spontaneous reactions. Reactions that are endergonic, when energy must be supplied, these are called non-spontaneous. Now, spontaneous doesn't mean it happens automatically, right? This reaction isn't going to happen quickly, but it means energy is released over time. Non-spontaneous means energy needs to be put in. Okay, so negative delta G, so this negative Gibbs free energy means a change in free energy. There's been some change. And because we've had a release of energy, that energy that's released now can go on and do some sort of work. Okay, so how does this all connect to life? Well, remember, think back to the beginning of this video. Living things need energy to live. So how do we get energy to live? We've talked about this a little bit already, but basically we couple. Coupling means putting together exergonic and reactions that release energy with endergonic reactions that need energy. So if we need energy, we're going to first take energy from molecules and then use it somewhere else. So for example, let's say we take a polymer here, and this polymer has a lot of energy because we have these chemical bonds that have energy in them, right? High potential energy in these bonds, potential for those atoms to change position. Once those bonds break, now we have lowered the potential energy of these molecules, and we've released that energy to go somewhere else. That energy now is used to build new molecules. So we have these molecules, they're very stable, they have low energy. So in order to make a higher energy molecule, you need to input energy. It's like a math equation, right? So molecules that are more stable, molecules that are more disordered, molecules that have more entropy. In order to build a higher energy molecule, you need to input some energy in, right? So that is why we need to break apart molecules so that we can build new ones. All right, so how do we make sure that we have enough energy? Well, first we need to eat high energy organic molecules. So organic molecules are carbohydrates, lipids, and proteins. So we eat food. Then we break them down, we digest them through catabolism. Catabolism is breaking apart molecules. And then through the catabolism molecules, we capture energy in the form that a cell can use. So remember how catabolism releases energy? Well, where does that energy go? Well, we need that energy to be in a form that can be used by the cell. We need some sort of energy currency. So the glucose molecules, the fat molecules, the protein molecules that we eat, it's not actually, those molecules aren't able to be quickly used as energy by the cell. We need a way to actually move energy around quickly in smaller pieces. And that is what brings us to ATP. So ATP is a molecule that basically is the energy currency for the cell. So if you think about glucose, it's kind of like a really high valuable molecule. It's kind of like $100 bill, right? It's got a lot of value. It's got a lot of energy in it. But that's so much energy, it can't be used in all one place, right? If you go to the dollar store and you want to go buy some things, you're not going to use a $100 bill. You need a smaller form of currency to spend in multiple places at once. So we take that high energy glucose and we create smaller molecules called ATP. But what is ATP? ATP is a molecule that looks like this right here. And it stands for adenosine triphosphate. Adenosine is the A, tri, the T, and phosphate is the P. And it's basically a nucleotide. So think back to our organic molecules, where nucleotides, for example, DNA and RNA are nucleotides, but ATP is also an example of a nucleotide. And nucleotides are made up of sugar, a phosphate, and a base. So this is RNA nucleotide, but ATP looks a little bit different. ATP now has a ribose sugar here, an adenine base, so that's the base, A. If you think of in DNA or RNA, it's an adenine. That's why it's ATP, adenine. And then you have phosphate groups. So here's one phosphate, so the phosphorus, and then 1, 2, 3, 4 oxygen, phosphorus, 1, 2, 3, 4 oxygen, phosphorus, 1, 2, 3, 4 oxygen. So there's 3 phosphate groups. This is 1 phosphate functional group, 2, and 3. Now, you can actually modify this a little bit. So let's say you just have the adenine and the ribose and 1 phosphate. We call that adenosine monophosphate. Let's say you take that adenosine monophosphate, so that would be just this, this, and this, and you add a phosphorus. We call this inorganic phosphate. Basically, if the phosphate group is by itself, we call it inorganic phosphate. So if you were to take AMP, adenosine monophosphate, and add another phosphate group, now you have adenosine diphosphate. Di means 2. If you were to take the adenosine diphosphate and add another phosphate group, now you have 3 phosphates, adenosine triphosphate. Those are 3 phosphate groups here. And then it forms this molecule called ATP. Right? So here's the ribose sugar, here's the adenine base, and 1, 2, 3 phosphate groups, ATP. So we call ATP an energy storing molecule. Okay, now here we have our sugar, our base, and our phosphate. This is AMP, adenosine monophosphate. But notice here that this phosphorus is bonded to these oxygen atoms. And these oxygen atoms are extremely electronegative. They're actually negatively charged here. So if you wanted to add another phosphate group, notice that now these negative charges are repelling each other. It's really, really hard to add on a new phosphate group here because now these oxygens are repelling each other. And this is not stable. This is a very unstable molecule. This can break very easily. But there's a lot of energy stored in this bond. Think about it. It's kind of like a spring that's loaded now, right? So now that you have 2 phosphate groups, those oxygens, those negative oxygens are pushing against each other. And as soon as it gets released, boom, now you have released a ton of energy because there's like so much potential energy stored. These bonds, these molecules are not happy with each other. They're very unstable. So they have the potential to release a lot of energy. So the first bond has some energy. Then the second bond, if you were to now bond 1, 2, and 3 phosphate groups, now you have 3 phosphate groups that are now they have a ton of energy. They want to push against each other. And the most energy is stored in this third phosphate group. So the delta G, if you were to release that phosphate off, you would have an overall release of 7.3 kilocalories per mole. That's just a unit of Gibbs free energy, the amount of energy that gets released. So because these phosphate groups are so closely packed, it's kind of like a spring loaded. As soon as that gets released, there's so much energy that comes back. And again, here's the explanation of why. So here's our sugar, here's our base, our adenine base, and then here's our first phosphate group. So again, these oxygen atoms, they do not like each other. They're repelling. And so that repulsive force, now that's a strong force that's going to want those molecules to separate and release a lot of energy. So again, we have really unstable bonds here. So ATP is an excellent energy donor, kind of like we just talked about. So how does ATP transfer energy? So if you have this ATP molecule, here's your sugar, here's your base, here's your phosphate groups. A process called phosphorylation is the removing or addition of this phosphate group here. So when ATP is ready to transfer energy, how does it do that? Well, it takes this third phosphate group and basically you break this bond right here. Okay, so now you're releasing your third phosphate from the second one. And remember, that had a ton of energy. And so you're actually going to release a delta G. The available energy is 7.3 kilocalories per mole. Or another way of thinking about this, another unit of energy is 30 kilojoules per mole. So it's negative because that's the overall net release of energy. So that's why it's negative. Started off with 7.3, and now that amount is released from the reactants to the products. Now, you can think of this third phosphate group as kind of like imagine like this ATP molecule is really, really upset with this last third phosphate group. And it basically wants to get rid of it, right? These phosphates, they're not happy with this last one. They want to get rid of it. They're repelled from it. Kind of like if you were to think about someone in a relationship and let's say they're in a toxic relationship and they want nothing more than to get rid of their partner, right? So what they're going to do is they're going to try to, as much as they can, to release this phosphate. Now, when this phosphate group gets released, it can latch on to other molecules and make them more unstable, kind of like this molecule is unstable. If this now gets attached to a new molecule, those negative oxygen atoms are going to destabilize other molecules. And that addition of the phosphate group to other molecules now supplies it with a lot of energy. Okay, so why don't we just eat ATP then? Why do plants have to build glucose? Why can't plants just take energy from the sun and build ATP molecules? Well, the problem is ATP is such an unstable molecule. It's so reactive, right? Remember those 3 phosphate groups, they have so much energy stored in them that it doesn't actually exist for very long amounts of time. So that inorganic phosphate, that third phosphate group actually comes off really quickly. And that's good because that removal of that last phosphate group supplies energy for cellular work, right? So that inorganic phosphate, remember, will go latch on to something else and now destabilize it and supply it with a lot of energy. But then this phosphate group can actually be re-added to adenosine diphosphate, ADP, and now you've regenerated ATP, right? So in order to do that, in order to take ADP and add phosphorus, these 2 molecules are more stable here, right? The ADP, it's more stable, it's more chill with just 2 phosphate groups. Adding a whole nother phosphate group, that's going to make it even more unstable or have even more energy. So in order to do that, you actually need energy to input and to turn ADP back into ATP. But your muscle cells can do this. Your muscle cells can regenerate ATP from ADP. And this process is called cellular respiration. So the reaction that takes ADP and forcefully adds back on that phosphate. Remember, this is like that toxic partner that is just going and wreaking havoc in other places. In order to get that back on the train, we're going to need some energy. That's called cellular respiration. Okay. So ATP is just a short-term use of energy. If we want to store energy for the long term, like let's say we want to save energy, we just store that energy in carbohydrates and fats. So carbohydrate can hang out for a while. Carbohydrates are pretty stable. And so if you wanted to get energy from that carbohydrate or that fat, you could break it down. You could do cellular respiration and break it down, and then get ATP from it. And so your working muscles actually recycle 10 million ATPs per second. So this process happens really, really fast in your muscles. But then if you want to store that energy long term, you're going to store that energy in more stable molecules of carbs and fats. So thinking back to metabolism, thinking about organisms getting energy, what's the whole point? Why do we need to eat? Why do we need to metabolize? What's the whole point of all these chemical reactions? Well, the main point is to make ATP. So in our cells, we have this very special organelle called the mitochondria. This mitochondria is found in plant cells and animal cells, all eukaryotic cells. And these mitochondria, their job is to take glucose molecules, take larger, a little bit more stable molecules, and break the bonds of glucose molecules. And in that process, the breaking of those glucose molecules, you're releasing energy. And that release of energy actually goes to and make ATP. So let's take a look a little bit more closely and put this all together. So earlier in this video, we talked about exergonic reactions. So let's say we take this glucose molecule, we break it apart, and this is called cellular respiration. Basically, in the mitochondria, we take a glucose molecule, we break it apart. We take a really organized, high energy molecule, we break it apart, and we increase the entropy. We also make more stable molecules here, right? They're more stable molecules because they're more spread out. There's less potential for them to change their position. But where does that energy go? Well, that energy now is going to go and help make ATP. We'll come back to that in a second. And then we have some waste products here. So after we break apart glucose, now we have some molecules laying around. What are those molecules? Well, those molecules are now carbon dioxide and water. Those carbon dioxide molecules are just waste products. They're waste. The whole point was to break apart this glucose so that we could release energy to go make ATP. And then now we have these waste products. And so for organisms that do cellular respiration, those waste products are now going to be released. For us humans, these carbon dioxide molecules will be released through our breath, and the water will be basically used by our cells or released when we go to the bathroom. So we talked about this release of energy, right? There's overall some available energy that gets released here. So what does it do? So that energy that gets released is energy that basically adds this inorganic phosphate to our ADP molecule. Because remember, this is really hard to do. It's really hard to put that inorganic phosphate back on the ADP. The ADP is more stable than ATP, right? So as soon as you add that inorganic phosphate, now those oxygens in the phosphate are going to push up against the oxygens in the second phosphate here in the ADP. And so that's hard to do. So it requires energy to build this molecule. So ATP synthesizing, making ATP from ADP and inorganic phosphate requires an input of energy. Where does that energy come? It comes from catabolism. It comes from chopping up your glucose molecules. These exergonic reactions, right? So going back here, this exergonic reaction, chopping up glucose, releasing energy. Now you can create ATP. Then remember that ATP, it's a high energy molecule. It's very unstable. It has a lot of potential energy, right? If you complete ATP hydrolysis, so you use water to phosphorylate. Phosphorylate means chop off that third phosphate group. Now energy gets released to go do work. So now you can build new molecules. You can complete endergonic reactions, reactions that need energy. That energy comes from breaking that last phosphate group. And then the cycle repeats and repeats. So what happens once you generate ATP? Well, ATP can be used to do work in the cell. So thinking back to Unit 2, when we're thinking about membrane transport, So if you're going to move a molecule against its concentration gradient, right, lower concentration of molecule here compared to outside the cell, you can use these proteins to basically pump the molecules out, but you're going to need some energy to do that. So the way to do that is you take ATP, you phosphorylize it, so you cut off that last phosphate group, and that phosphate group will now bond to this protein here, destabilize the protein, and supply it with energy, and that supply of energy is going to cause the protein to change shape and basically shuttle these proteins of solute out of the cell. Then that phosphate will be released, and now you can regenerate ATP again. So ADP molecules will pick up that phosphate group, and now you have ATP again. And so this is why we need ATP, to push molecules against their concentration gradient. Okay, now that we've learned a little bit more about ATP and how ATP is used to supply our cells with energy, now we're going to transition into looking at how chemical reactions occur with the help of something called enzymes. So again, we can basically summarize metabolism by naming two types of chemical reactions: synthesizing or making molecules or forming bonds, or digesting, hydrolysis, breaking bonds. Now, in order to do this doesn't always happen on its own. Yes, eventually dehydration synthesis would bond two monomers to make a polymer, but it would happen over a really long period of time. And enzymes actually have the special job of helping these two molecules come together, right? It's kind of like a matchmaker. Or sometimes these enzymes are kind of like divorce attorneys. They help two molecules or two subunits of a polymer break apart, right? They kind of speed up either breaking or the making of molecules. Now, enzymes are examples of proteins, and proteins have so many different functions in the human body. So you can take a look here and look at some of the examples of different type of proteins. Remember, cells are basically protein factories, and the cells can make all these different types of proteins. But for today, we're going to look at enzymes, and enzymes are also called catalysts or biocatalysts, and they're used for almost all of these chemical reactions that happen in the body. So metabolism needs enzymes to occur, and we'll look at why. But remember, enzymes are simply proteins. And we think back to Unit 1, proteins are just a bunch of amino acids, these monomers, connected through covalent peptide bonds, like beads on a string. And when these primary structure proteins bond together, when these nearby adjacent amino acids form hydrogen bonds with each other, they form now our secondary structure, beta-pleated sheets and alpha helices. This is our secondary structure. Hydrogen bonds hold together these structures. When these beta-pleated sheets and alpha helices bind together as well, there are bonds between these two. That forms our tertiary protein structure. And this is like one subunit. If we were to put multiple subunits together or multiple of these amino acid chains together, then you would find our quaternary protein structure. So again, proteins, one type of protein is an enzyme. Now, enzymes help speed up chemical reactions. So if you were to take a molecule called hydrogen peroxide and you were to break it apart, eventually it would actually be able to create water and oxygen. So the chemical formula for hydrogen peroxide is so there's two of these, H2O2. So there's two hydrogens, two oxygens in one molecule. If you had two molecules of them, then you'd have four hydrogens and four oxygens. If you were to break this molecule apart, it would take a long time. So you use an enzyme called catalase. Whenever you see the ase at the end, that tells you it's an enzyme. Ase always as the suffix for enzymes. Then you would have, so again, we have four hydrogens on this side. Notice here now we have four hydrogens on this side, four oxygens, two here. So there's two here and two here, four oxygens on this side. So you've just broken apart hydrogen peroxide and you've made two smaller molecules, right? You've cut this down, you've broken it apart, but that requires an enzyme called catalase. So how do chemical reactions use enzymes? Well, we've kind of talked about this in our case study, our Great Dane, or our case study on the dog in class. But basically, chemical reactions have an energy barrier called activation energy. So if this is our reactant, there's going to need to be an input of energy in order for the reaction to start. And remember that reason for that is because if you have a reactant, so you have a molecule with atoms that are bonded together, in order to break those bonds, you actually need some energy to put in first. Because the molecule is pretty stable. The atoms they're just fine. They're chilling. And so in order to break that, you kind of need a little bit of energy to get that molecule or those atoms kind of separated from each other. But as soon as they start getting separated, that releases a lot more energy. And so overall, you have a net release of energy. But because there's some energy that's needed to be put in first, this actually could take years to happen spontaneously. This would take years to happen before you make a product. And so, in order to lower this activation energy, or EA, we're going to need to use an enzyme. So, again, enzymes are proteins that act as biological catalysts. Catalyst means to speed up. They speed up chemical reactions. So, for example, if we take a sugar called lactose, whenever you see the ose at the end, that tells you it's a sugar, like glucose or fructose, lactose. You could take an enzyme called lactase. So a very specific enzyme will help break apart lactose, right? And dehydration synthesis, so it requires some water to break these bonds. And now you have the two smaller molecules. So now you have galactose and glucose. These are monomers that originally made up lactose, but now you broke them apart. Now, in order to do this, you actually need an enzyme. So an enzyme looks like this up here, and you'll take your lactose molecule. The lactose molecule will enter in this part here of the enzyme called the binding site. The enzyme will kind of close around it, and the chemical reaction will happen, and then it will open up again, and now you have your products. So your reactants come in, your products come out. So how do enzymes speed up chemical reactions? Remember, if this is our amount of energy that we need, as our base level, our reactants, there's some energy that needs to be put in to break the bonds of the molecule, and then energy overall gets released. But without this activation, without the enzyme, the activation energy would be very high. This would take a long time. A lot of energy would need to be put in. Now, how could we speed up, or how could we input energy? Well, one way to input energy into a reaction is if you heat it up. So if you heat up the reactants, that actually puts energy into the reaction. And that could actually speed up the reaction. But the problem is, if you heat up molecules, sometimes it causes them to heat up too much, and it can actually break apart or denature the proteins. And so that would actually be dangerous in the human body if we added too much heat. So instead of heating up, we can actually just lower the activation energy. So there's less energy needed to break apart the bonds of the original reactants. All right, now this is overall an exergonic reaction because we're releasing energy. So if these are our reactions A, B, C, D, and these are our products A, C, B, D, you're going to need some energy put in at first in order to break these bonds, right? Because again, these atoms right here, they're pretty stable. Like let's say this is a more positive and this is a more negative atom. They're bonded together, right? They're sharing electrons. So in order to kind of disrupt their chill state, you're going to have to input some energy. But once you do, they're going to release, they're going to move away from each other, release energy, and now new bonds can form. But overall, we have a net release of energy. Delta G is negative. So again, where did the reactions get energy to overcome this activation energy? Not from heat. Heat could be an input of energy. Heat could also break these bonds apart, but that is not realistic in the human body. Heat could actually ruin molecules and enzymes in your body. So what's actually happening when enzymes speed up chemical reactions? Well, here's our enzyme here in purple. And here is our reactants. Here's the thing that's coming into the chemical reaction. And this, another word for it is called a substrate. So a substrate is the reactant, the thing that's going into a chemical reaction. The enzyme is the catalyst, the thing that's speeding up the reaction. And the enzyme will actually have this spot here called the active site. And the active site has a specific shape that can bond to a specific substrate. So one substrate can fit into one enzyme. So there's thousands of different types of enzymes, and each enzyme is specifically designed to fit one specific substrate. And so this site here is the catalytic site. So this is where the substrate can bond. And when it's bonded, this enzyme can actually speed up some sort of chemical reaction. So for this specific enzyme, it's actually going to break the reactants and form products that now are monomers. And then that enzyme can now be recycled and used again. Now, sometimes when we want to simplify this, we can call it the lock and key model. So scientists used to think that the substrate kind of acted like a key and it went into like a lock here. But this is a very simplistic model. So if you were to look at an example of this lock and key model, you would see this substrate. So let's say the reactant is a sucrose molecule. It's got a glucose and a fructose, but it's bonded together. So it's this polymer right here, this disaccharide, two sugars, right? So if it were to enter into our enzyme, now we have our enzyme substrate complex. Enzyme substrate complex means you have both of them together. So the substrate will bind. Now the enzyme, remember, it's specifically designed to only allow sucrose, this sugar, to get in. We call it sucrase. Ace means enzyme. Now, with the addition of water, right, we are now hydrolyzing the molecule. We're inputting water to break the bonds. Now we have converted the substrate into our products here. We've broken sucrose down into glucose and fructose, right? And then now the enzyme will release the products, and now we can add in another sucrose molecule. So how does the substrate get into the enzyme? Well, there's actually some small weak bonds. So some hydrogen bonds will allow the substrate to bind to the enzyme. And we kind of think of it as like a key fitting into the lock. So there's one key that fits in one lock, right? If the wrong substrate comes up to the wrong enzyme, it won't fit. Okay, but this is not the best model of enzymes. What it really is more like is something called induced fit. So let me explain. So if this is our enzyme here, here's our substrate, here's our reactant. The substrate will enter into the active site. It will bond hydrogen bonds here. So the atoms that make up the substrate will bond to the active site through hydrogen bonding here. And then instead of it being like a key, what actually happens is more like a handshake. So when you shake someone's hand, your hand kind of molds or fits around someone else's hand. So think of it as kind of like the enzyme's going to like hug onto the substrate. It's going to kind of mold and form like into the substrate. So it's not. So if you think of a lock and key, the lock and key model isn't great because the lock doesn't fold and mold around the key. But what really happens is the enzyme will actually fold around the substrate here to form the enzyme substrate complex. So here, let's look at how it actually happens. So the enzyme here has an active site and it's empty. When the substrate comes along, sucrose, the sucrase, the enzyme, will bind to the substrate. And this is called induced fit. Induced means because it fits because something is changing. It's kind of hugging around. And with the addition of water, now we can now break apart the bonds of the sucrose, right? With the addition of water, hydrolysis, and now we've converted the substrate to the products, fructose and glucose, and now those products are released. But the reason why we were able to speed up this chemical reaction is because that enzyme actually kind of squeezed the sucrose molecule, and that squeezing of the sucrose molecule is able to help break those bonds even faster. And so then once those glucose and fructose are released, the active site is now open again and a new substrate can bind. So the lock and key is more like induced fit. The enzyme has a conformational change. Conformational means the actual shape of the enzyme changes. It wraps around the substrate, catalyzing, speeding up the reaction. So let's think back to what we learned about activation energy. So remember, there's a certain amount of energy that needs to be put in order to break apart this molecule. This molecule is pretty stable. So in order to break this bond, you need some energy put in. So what is that energy put in? Well, what actually happens here is this protein is going to stretch the substrate or put some bend into the substrate, helping to break these bonds even faster. So literally, the enzyme actually acts as like this kind of like hand that grabs and puts pressure on the substrate. Another way that the catalyst can lower the activation energy is they can actually provide a small microenvironment. So this active site here can actually supply like some sort of different environment, kind of like a little cave, where it provides the right environment. So for one example, like maybe it's the correct pH. So a small environment inside here, the right pH that's conducive to a faster reaction. Okay, so that's how catalysts lower the activation energy. It's because this change of the protein either puts pressure or creates the perfect environment for this reaction to occur. So, two ways that enzymes can work, two ways that catalysts can work. They can either take the substrate, and as the enzyme squeezes or conformation change means the enzyme literally changed shape. It squeezes around it, it hugs the substrate, can actually help break those bonds. Or the enzyme can actually help make the bonds. So as the substrate binds to the active site, it can actually, as it squeezes those substrates, it can actually push those substrates together and form bonds, helping to make a product. So the enzyme can either act as a matchmaker, taking two monomers and making a polymer, right? Dehydration synthesis using with the release of water here, or it can act as like a divorce attorney. So taking a polymer or a disaccharide here, and as it enters into the active site, the enzyme will change shape and it will kind of squeeze and tug on this substrate and it will release a product. But now that product is now has two monomers that are released. It's breaking the molecule apart. All right, so just something to note. The enzymes that we're looking at here are reaction specific. So every enzyme only works with a specific substrate. So if you have substrate A, B, C, D, there's all different molecules, but you only have this one enzyme. Notice that the active site has a certain shape. Literally, this protein here has bent, it's folded, its quaternary structure, it's all the amino acid chains has shaped it into this specific shape, right? That's why folding of proteins are so important. Each protein forms into a specific shape. And what this does is it only allows a certain substrate to fit in. And so only the substrate that has a certain shape is able to form hydrogen or ionic bonds with this active site here. The chemical fit only fits this specific substrate. If this substrate would try to get in, it wouldn't have the correct shape to get in. It couldn't form hydrogen or ionic bonds to fit and stay in place in this active site.